Hello student. In our previous video, we have discussed about the biogas generation and also we have discussed about the from what we get the biogas, that is a which acts as a biomass, that is a generally we have considered the cattle dung as well as piggery waste and poultry dropping. These all are also used for the biogas generation, and after that there is a agrochemical waste material, that is a crop residues and some other materials like a kitchen waste, paper waste, waste from sugarcane refinery. These all are the main sources of obtaining the biogas, that is these sources are used for the biogas generation. And also we have discussed about the process which is the digestion process, or simply we call it as a anaerobic digestion process. And in that again there are three phases, that is enzymatic hydrolysis, and second stage is the acid formation, and last one which is the third, that is a methane formation. These three main phases which are comes under the digestion or anaerobic digestion. And in these stages there are acid formers and methane fermenter. These two main components which play important role in last two stages. And also there is a dynamic equilibrium between the acid former and methane formers is very important, because according to that the efficiency of the plant is decided. So this is all about the biogas generation we have discussed in our previous video. Now in this video we have to see the factors affecting biodigestion, and in syllabus which is given, that is only lists the factor, but we will discuss in brief. There are total 10 factors which affects the biodigestion. So we have to consider all these 10 factors. First factor is the temperature, second factor is the pressure, third factor is the solid to moisture ratio, fourth factor is the pH value, fifth is the feeding rate, sixth one is the carbon to nitrogen ratio and other nutrients in the biomass. Next one is the seventh one, seeding of biomass with the bacteria. Eighth, mixing or stirring. Ninth, retention time. And 10th, effect of toxic substances. These all are the 10 factors which affect the biodigestion. So we have to see one by one. So first factor is the temperature. The methane forming bacteria work best in the temperature ranges between 20 to 55 degree Celsius. As we mentioned in the previous videos, there are three phases which comes under the digestion process, and in methane forming process there is a methane fermenter, and this methane fermenter or bacteria, methane forming bacteria, work best in the temperature range, that is a 20 to 55 degree Celsius. Digestion at higher temperature proceed more rapidly than at lower temperature. That means the higher temperature is the best for the good digestion, and the procedure of digestion is higher at higher temperature and it lower at lower temperature. So high temperature is good for the any biogas plant or biodigestion process. Second parameter is the pressure parameter. A minimum pressure of 6 to 10 centimeter of water column, that is 12 bars, is the ideal for proper functioning of the plant. That means the pressure range which is lies in between 6 to 10 centimeter of a water column, or simply which is the 12 bars, is the ideal pressure for any plant for good operation, that is a proper functioning of the plant. So this is the pressure parameter which affects the biodigestion. If it is improper, then it affects the biodigestion process. Third parameter is the solid to moisture ratio. Solid to moisture ratio, that means the consistency of the slurry in the biogas plant is a proper. For that we add some quantity of water. For various input materials, the optimum ratio of solid to moisture should be adjusted by mixing extra water for the best result. In order to obtain the best solid to moist ratio, we added some water to the slurry and we get the proper concentration or proper soil to moisture ratio. Then we get the best result from that plant. Fourth parameter is the, or fourth factor is the pH value. We know that pH value varies from 0 to 14. When the pH value lies between 0 to 7, then the substance having nature, that is acidic nature. When it is 7, then the substance is a neutral, and when it is above the 7, then the nature of the substance is the basic. We know all this basic part. During CH4 formation stage, pH value of 6.5 to 7.5 is maintained. When there is a formation of methane, then we have to maintain the pH value, that is 6.5 to 7.5. Next parameter is the fifth one, that is a feeding rate. The uniform feed rate is maintained. That means the providing of the slurry or biomass to the biogas plant, that is a feeding rate, and this rate is uniformly maintained for the proper functioning of the biogas plant. The faster feeding rate will not help increase the gas production. When the feeding rate is faster, then there is no any useful, because it does not help to increase the production of the gas. It must be, or it should be, uniform feed rate. So this is the feed rate. Next parameter is the, or next factor is the sixth one, is the carbon to nitrogen ratio and other nutrients in the biomass. The optimal carbon to nitrogen ratio required for maximum microbiological activity is 30:1. We know that the carbon element and nitrogen element present in the biomass, and the ratio of these two quantities is 30:1, and this is very important for the microbiological activity in the biogas plant. For optimal growth and activity of bacteria, it is essential that required nutrients are available in a correct chemical form and concentration. We know that there are different anaerobic bacteria or methane fermenter or acid former. For the growth and activity of those bacteria, there is a very important part, that is the proper concentration of the nutrients available in the chemical form. That means when there is a proper concentration of the nutrients available in the biogas, then or the in a digester, then and then the growth of bacteria and activity of bacteria is proper, and we get the better efficiency of that biogas plant. This is the sixth one, that is a carbon to nitrogen ratio and other nutrients in the biomass. Seventh is the seeding of the biomass with the bacteria. That means when we construct the new biogas plant, then we can add slurry, but the slurry is a fresh slurry. For the fermentation, to start the fermentation process, we have to add some bacteria to the freshly charged plant. So the seeding is an important process. To start and accelerate fermentation process, small amount of digested slurry containing methane forming bacteria is added to the freshly charged plant. This is known as a seeding. That means the addition of the digested slurry to the freshly charged plant to accelerate and start fermentation process. So this process is known as the seeding process. That means feeding of the digested slurry to the freshly charged plant, and all this process to start the and or to accelerate the fermentation process. So the next process will take place. So this is the seeding of the biomass with the bacteria. Next parameter or next factor, 8.1, is the mixing or stirring. Mixing or stirring is a very important factor, because it maintain the uniform concentration of the slurry or the material which is present in the digester or the chamber. Mixing prevents the deposition of solids at the bottom and it minimizes the formation of scum at the surface. We know that when there is a slurry, then there is a formation of scum at the surface, and due to scum formation there is not uniformity in the slurry. So for the breaking of that scum at the surface we require external stirrer, and due to stirrer the scum will break and we get the uniform concentration. Then and also the stirrer or mixing prevents the deposition of solid material. In this slurry there is a solid material and after some time it get deposited at the bottom. When we external stirrer is used, when we use external stirrer, then this solid material does not deposit at the bottom side, so the concentration will be uniform. It maintains uniformity in the concentration. Who? Stirrer. That means stirrer helps to break this scum at the surface and it also prevent the deposition of the solid material at the base of the plant or at the base of the digester. Next parameter is the retention time. Retention time is the time duration for which the slurry remains in the plant or the time that is available for biodigestion. That means retention time is the time for which the slurry remains in the tank or digester. This time interval or this time duration is known as a retention time. That is a total time for the biodigestion for the slurry which is present in the digester. This time is known as the retention time. Retention time is also very important for the biodigestion process. When it varies according to the plant and it also affect to the efficiency of the plant. Next and last one factor is the effect of toxic substances. As we mentioned already, the biomass is used from the variety of field, such as agricultural field, sugarcane factory, food waste, kitchen waste, cattle dung. All these generally the waste material from the kitchen waste and also from the household uses and also from the industrial waste. These material consist of different toxic substances and these toxic substances hazardous or toxic to the bacteria which is present in the slurry or in the digester tank, so that the efficiency of the plant gets decreases. So this is very important factor for the activity and growth of the microbes or bacteria which is present in the slurry or which play very important role for the biodigestion process. High concentration of ammonia, antibiotics, pesticides, detergents, heavy metals like chromium, copper, nickel, zinc, etc. are toxic to bacteria responsible for biodigestion process. As we mentioned, there are variety of toxic substances, that is a high concentration of ammonia, antibiotics from the medical field, pesticides that from agricultural field, detergents from the household use, heavy metals like chromium, copper, nickel, zinc from different industrial areas. All these materials are toxic, and when we use this material as a raw material or the biomass for the production of the biogas, then after start the fermentation process, these all material are hazardous to the bacteria which are responsible for biodigestion. Sometimes the bacteria may damage due to all these toxic substances, and so we get the minimum efficiency of that plant. So this factor play very important role for the efficiency of that particular plant. So all these are the factors affecting for the biodigestion process. First factor is the temperature, second is the pressure, third one is the moisture to solid to moisture ratio, next one is the pH value, feeding rate, carbon to nitrogen ratio and other nutrients in the biomass. Seventh one is the seeding of biomass with bacteria. eighth one is the mixing or stirring, next one is the retention time, and last one is the effect of toxic substances. All these factors affect the biodigestion process. So the name given is that factor affecting the biodigestion process. So this is all about that, all about these factors. So for the biodigestion process we have to take into account all these factors, because from that we get the efficiency of the biogas plant. So this is all about the factors affecting the biodigestion. Now in our next video we will discuss about the working principle of the biogas plant, that means how the biogas plant works, different parts of the biogas plant, and also how the biomass is converted into the biogas or biodiesel. These all are comes under the working principle of the biogas plant. So in our next video we will discuss about the working principle of the biogas plant. So in that video we will continue with that.